So to start, so when we say pharmacokinetics, we usually abbreviate this as PK. This is concerned with the study and characterization of time course of drug, ADME. So again, when we say ADME, this refers to the absorption, distribution, metabolism, and excretion of the drug in the body. So this is also concerned with the relationship of these processes to intensity and duration of the drug effects. So for example, the time that the substance or the drug stays in the body, the longer would also be the duration of the action and the effect of that particular drug. So it has been described as what the body does to the drug. And so when we are going to compare that with PD or the pharmacodynamics will answer the question what the drug does to the body or the power of the drug to the body. For the PK or the pharmacokinetics, it will answer the question what the body does to the drug. So for the different components of the pharmacokinetics, so it is composed again of absorption, that is the first step, followed by distribution. Then the third would be metabolism or biotransformation, and the excretion or the elimination. So for one of the categories of pharmacokinetics is the clinical pharmacokinetics. So when we say clinical pharmacokinetics, from the word clinical, that is the use of PK information to select and define rational drug therapy in clinical patients. So for example, what are the PK informations that we can use in order for a drug to be used in the treatment of certain ailments. So for example, the PK information that can be considered are the half-life of the drug, that is one of the parameters that we can use. Another is the withdrawal period or the withdrawal time of the drug. So these particular parameters can be used in order for us to be able to choose a drug that is safe and that is free of detrimental adverse effects to the patient. So these are the components of the pharmacokinetics and the pharmacodynamics. And this schematic diagram will illustrate the relationship between PK and PD. So of course, we start with the administration of the drug. The drug administration can either be oral or parenteral. So after the drug administration, the drug would undergo absorption, distribution, metabolism. Then after metabolism, it can undergo elimination. So during the drug distribution process, the drug itself, again, when we say drug distribution, that is the transport of the drug to the different tissues in the body, which contains the specific receptors, specific target elements that are found within that particular organ. So the drug, when it is present in the blood, it will be distributed to different tissues in the body. It will be distributed to different fluid compartments in the body, such as the intracellular fluid and the extracellular fluid. So when it is present in this particular compartment, it can act or it can be acted upon by different regulatory proteins. So the drug could either act on a receptor, an enzyme, an ion channel, or a transporter. So for the transporter, examples of the transporters are our plasma proteins that are present in the blood, or there are also transporters or carrier proteins that are present in the plasma membrane or the cell membrane. So the regulatory proteins, so the drugs can act, for example, on the receptor that are found within the target tissue. Of course, for the drug to be able to interact with the receptors, it must have first the affinity or the ability to bind to the receptor in the target tissue. So after the drug is able to bind to its target tissue, that is the time that it can manifest its mechanism of action or MOA. So after binding, the drug could either induce an effect or not induce an effect, depending on the property of that particular drug. So the other components of the pharmacokinetics in this diagram is the different routes of drug administration. So we can, for example, administer a drug through ingestion, by way of the oral route, or we can use injection. For example, we can use intramuscular, IM, SC, subcutaneous, or intradermal, through the dermis or through the peritoneum, through intraperitoneal. So for those drugs that are administered through the oral route, they can undergo absorption in the stomach, and they can be present in the bloodstream. So in the bloodstream and in the GIT, they can undergo metabolism. So after absorption, they can undergo distribution to the different tissues such as the liver. So in the liver, the drug can undergo metabolism there, in the liver, and after that, it can be excreted in the body. For example, when a drug will be metabolized in the liver, it can undergo this what we call the first pass, a first pass effect in the liver. The metabolites of that drug metabolism could either be distributed. It could be further distributed in the bloodstream to act on the target tissue, or the other metabolites, those metabolites that are inactive, could now be excreted in the body. So that is under the pharmacokinetics. The drugs that are present in the bloodstream could also be filtered. It will be transported to the kidney, where it can be filtered, it can be reabsorbed, and it can be secreted in the kidney. So those drug metabolites that are filtered and that are secreted in the kidney, particularly in the nephron, will be excreted through the urine. So the other drug metabolites that are present in the blood will also undergo tissue uptake. It can reach the target tissue to act on the receptors. So again, the receptors in the tissue would either be located at the cell surface, or it can either be located in the cytoplasm or in the nucleus. So for a drug to be able to move or to diffuse across the cell membrane, they must have this certain kind of characteristic in order for them to be able to interact with the receptors. So for example, for those drugs that that needs to enter the cell in order to interact with the receptors, so they must have this certain kind of lipid solubility in order for them to enter the cell and to act with the receptor and to activate certain enzymes in order to produce its biological activity. So the time from the binding of the drug to its target tissue up to the time that the drug will manifest its effect on the target tissue is known as the pharmacokinetics of the drug, and the time from the administration of the drug up until the time that the drug will be excreted in the body is known as the PK of the drug. So why do we study PK? So scientists study the PK of the drug to achieve the following. So the first one is the study of the PK is very important in order to determine the onset, the duration, and the intensity of the drug effect. So when we say onset, that is the time from the administration of the drug up to the time that the effect of the drug would be observed, no, in the animal or in the body. The duration is of course the time, no, from the administration up to the time that the drug effect will start and up to the time that the drug effect will subside or will wane in the body. So for the intensity, we have those drugs that are more effective than the other. We also have those drugs that are more potent compared with the other drugs, now depending on different types of parameters. So we also study PK in order to enhance the efficiency of drug delivery. We also study PK in order to reduce the risk of toxicity to the patient during drug therapy. So a component of the study of the pharmacokinetic is that we have to consider the different adverse effects that the drug can produce, no, in the different biological systems. So that is a component of the PK. So knowing the toxicity, no, the risk, the toxicity, and the adverse effects of a particular drug could help the clinician to choose the appropriate drug that is suitable, that is safe for the patient. So in the study of pharmacokinetics, we have different factors that we have to consider. So of course, in choosing the right drug, that we are going to use, we must consider the efficacy of that particular drug, whether that particular drug is effective for a particular condition. Another parameter that we are going to consider is the lack of toxicity or the lack of adverse effect. So of course, in treating our patient, we need to balance whether that particular treatment or that particular intervention could be could produce, a more benefits compared to the risk. So if the risk outweigh the benefit, so the treatment would be useless. So the drug, that we are going to choose should have a minimum effect and maximum, efficacy at an optimal dose. So when we say dose, that refers to the amount of the drug, that will be given to the patient at a particular time. So when we say optimal dose, that refers to the lowest dose that will produce the least toxicity at an optimum efficacy or at uh, optimum efficacy, to the patient. Another consideration for food animal veterinarians or for those veterinarians that work with food producing animals, such as poultry, um, swine, and cattle, is that they have to consider the residues, the residues that are present in the the byproducts of the food animals, such as meat, milk, and eggs. So during, the production phase of rearing or raising poultry, raising cattle, and raising swine, we usually use antimicrobials and other chemotherapeutic agents in order to treat different types of diseases. So along the way, we use certain agents in order to in order to treat different types of infections different types of diseases. But when we are not going, to follow the recommendations as to the withdrawal period or the withdrawal time of the different drugs, so it could lead to the uh, manifestation or to the manifestation of the antimicrobial residues in the meat, milk, and eggs. So when we are not going to follow, the recommended withdrawal period of a particular drug or a particular antibiotic, so that could lead to the development of the antibiotic residue in the product. For example, when we are going to use an antibiotic to treat a certain type of infection, and we do not follow the minimum withdrawal period. So when we say withdrawal period, that is the time from the administration of the drug up to the time that the level of the antimicrobial in the meat, milk, and egg is within the tolerable level or within the safe level. So when we are not going to consider that, and when we are going to market the animal and we are going to slaughter the animal, the antibiotics that we have used to treat the diseases could still be present at a detrimental level to the meat, and milk and eggs. So this is one of the consideration of um, pharmacokinetics in the field of the food producing animals. We have to consider that the animal has undergone um, the proper treatment following the recommended withdrawal time or withdrawal period. So the components of pharmacokinetics are absorption, D for distribution, M for metabolism, and E for excretion. So these two parameters, the absorption and distribution, will influence the concentration attained in the immediate vicinity of the receptor sites. So for example, when the drug is well absorbed in the body and when the drug is well distributed in the body, so there will be more drugs that will be available in the uh, blood to act on the receptors. For metabolism and excretion, these two factors will be responsible for terminating the drug action. So when we say terminate, that is a factor or that is a process that will end, that will make the drug inactive in order for it to be excreted in the body. So when the drug is metabolized and when the drug is excreted in the body, so the concentration of the drug will also be reduced in the body, so that the duration of the effect will also differ. So for example, for those drugs with a shorter duration or stay in the body, their duration of their effect would also be shorter as compared to those drugs which are staying in the body for a longer period. So the next is the passage of drugs across biological membranes. So in order for a drug to act on its receptors, it must first diffuse, or it must first be transported along biological membranes in order for it to induce its effect. But first, we have first to consider the function in the anatomy of our biological membranes. So it is said that drugs must have some ability to dissolve in water to move around, be absorbed, and reach site of action. So this means that for a drug not to be able to uh, for a drug to be able to reach its site of action or its target tissue, it must have certain ability to dissolve in water in order for it to be transported, for example, in the blood. Drugs must also have a degree of lipid solubility to move around. The same, no? For a drug to be absorbed, it needs a certain degree of lipid solubility. So it is said that all cellular membranes are primarily lipid bilayers. So this means that all cell membranes are made up of lipid molecules and they are arranged in a bilayer. So when we say biological membranes, the other name of that is biomembrane or cell membrane. This is an enclosing or separating membrane that acts on a selectively permeable barrier within living things. So structures that define and control the composition of the space that they enclose. So we have two types of biological membranes. So those membranes that are found on the outer part of a typical cell are known as plasma membrane. So those membranes that are found forming the forming the outer component of the intracellular organelles such as the nucleus, the mitochondria, the Golgi, the lysosomes, and others are known as intracellular membranes. So the first attempt to describe the anatomy of the biological membranes was proposed by Singer and Nicholson in the year 1972. And this particular model is known as the fluid mosaic model. So this model provides a basic description of the structure and behavior of membranes in many cells. So according to this model, the plasma membrane is composed of the following components. So primarily, we have the phospholipids, we have the cholesterol, and we have the proteins. So they move freely and fluidly in the plane of the membrane. For the characteristic of the biological membranes, so the characteristic feature of it includes a bilayer of amphipathic phospholipid molecule that is oriented perpendicular to the plane of the membrane. So again, when we say bilayer, it is composed of two layers. When we say amphipathic, now this is a particular compound having both polar and non-polar groups. When we say phospholipid, now of course that is basically composed of a lipid molecule. So phospholipid is a type of a lipid molecule. So this is the diagram showing the fluid mosaic model. So the principal components are, so we have the lipids, that comprises the predominant part or the majority of the biological membrane. So this one here are all lipids, particularly phospholipids. We also have proteins. So the blue one are the proteins. So there are proteins that completely penetrates the cell membrane. There are also those proteins that are found attached to our cell membrane. We also have the glycoproteins. So when we say glycoproteins, these are proteins that are attached to the carbohydrates. We also have the glycolipids. These are lipid molecules that are attached to the carbohydrates. And within the cell membrane, we also have cholesterol molecules. So biological membranes are characteristic lipid bilayer. So they are arranged with polar and non-polar groups within it. Now, when we say polar, this refers to the hydrophilic heads that are present at both surfaces. So the exterior and the interior surface of the cell membrane. And The interior part no, of the lipid bilayer is basically composed of the tails, which are described to be hydrophobic in nature. And the lipid bilayer is also composed of aqueous channels or integral proteins that act as transport system. So again, when we say aqueous channels, these are channels that will uh, act as a transport system for water. They will act no, as a transport system for water because water no, cannot freely diffuse across the membrane. So again, the lipid bilayer is composed of a polar group no, and an nonpolar group. The polar groups are located inside and outside the cell. On between these polar groups are the tails, which are hydrophobic in nature. When we say hydrophilic heads, this refers to the polar head groups. And when we say hydrophilic, no, from the word hydro, water, philic, meaning affinity. So they have affinity for water. They are able to be wetted by water, or they are water soluble. So these are the water-loving component of the biological membranes. We also have the hydrophobic tails. So this is the nonpolar molecule or the nonpolar group of the biological membrane. From the word hydro, water, phobic, meaning fear, they are water-fearing, so they lack affinity for water, and they are unable to be wetted by water, or they are not water-soluble. We also have the term amphipathic or amphiphilic. So these are compounds that have both polar and nonpolar regions, giving them hydrophilic and lipophilic properties. So biological membrane, since it contains both polar and nonpolar group, uh, water-loving and fat-loving group, so they are called as amphipathic or amphiphilic. So these are the diagrammatic representation of a section of a lipid bilayer composed of the phospholipid. So these are the heads, and the heads contains the tail. The heads are said to be hydrophilic or water-loving. The tails are hydrophobic or water-fearing. So the phospholipid molecules are present in a bilayer. So we have two layers wherein the heads are located on the exterior and the tails are located on the interior. So this polar heads here is the water-loving portion of the biological membrane, and on the interior are the water-fearing component of the cell membrane. So when we are going to uh, consider the structure of a phospholipid molecule, so this phospholipid molecule will comprise now the main fabric or the main component of our biological membrane. So this one here would represent the head no, of the phospholipid molecule. It is hydrophilic and the tails are hydrophobic. So, so since this is a phospholipid molecule, no, a phospholipid molecule is basically composed of a lipid and a phosphate group. And so that this particular head here is being composed of the phosphate and glycerol. So glycerol is a basic unit of our lipids. Lipids are basically composed of two basic components. We have the glycerol and fatty acid. So the tail of the phospholipid molecule is represented by the fatty acid. And as we all know, no fatty acids can be saturated or non-saturated or unsaturated. So pag sinabi nating unsaturated, no, it is composed of double bond or double bonds. So pag mas maraming double bonds, so it would render the molecule weaker, no, as compared to a molecule having lesser no double bond. So in this example, no, there is a bending or kinking of the fatty acid molecule, no, because of the presence of our double bond. So therefore, when the biological membrane is mainly composed of a saturated type of fatty acid, so it would be more rigid as compared to a biological membrane having more unsaturated fatty acid. Okay, for the another no, illustration of our phospholipid molecule or our cell membrane. So this is made mostly of phospholipids. Now the majority or the major component of the membrane is phospholipids, and this form the bilayer. So this is another illustration of the phospholipid molecule containing the head and the two tails. So for those molecules that are hydrophobic, since our cell membrane is composed basically of a lipid bilayer, those molecules that are hydrophobic can pass freely to the lipid bilayer. When we say hydrophobic, these are molecules that are water-fearing, or they are nonpolar, or they are lipid soluble. When we say hydrophilic molecules, these are molecules that are water soluble, so they will not be able to dissolve in the lipid bilayer, and they will not be able to pass through the lipid bilayer. So example of a molecule that is polar or hydrophilic is water. So water cannot freely diffuse across the cell membrane, so that it needs a protein in order for it to be transported from one compartment to the other compartment. So the other composition of the biological membranes includes protein, carbohydrates that exist in a fluid state. So for the lipids, we have the sphingolipid and the glycerophospholipid. For the carbohydrates, so they are basically oligosaccharides in nature. They are attached to either lipid, forming glycolipids, or to proteins forming glycoproteins. So there are two general types of proteins. We have the integral and the peripheral. For the integral proteins, they are tightly bound to the membrane and inserted into or penetrate the lipid bilayer. For the peripheral proteins, they are loosely associated with the membrane through interactions with polar head groups or with integral membrane protein. So these are some of the examples of proteins that are present on the cell membrane. So for example, one of the functional class of proteins in the cell membrane are transporters. So when we say transporters, these are protein molecules that will transport, for example, ions or polar molecules into and out of the cell. So for example, we have the sodium pump or sodium potassium pump, or the sodium potassium ATPs. So this is another diagram of a plasma membrane showing the different components: the peripheral protein, integral protein, the extracellular fluid, that could refer to the blood plasma. These are the carbohydrates. The carbohydrates that are attached to the lipids are known as glycolipids. The carbohydrates that are attached to proteins are known as glycoproteins. And we also have the cholesterol. So one of the important property of biological membrane is that they are selectively permeable, meaning that they will not facilitate the diffusion or the entry of all types of cells, but they will only be selective for certain type of molecules. This is essential for effective separation of a cell or organelle from its surrounding. Another important property of the cell membrane is the fluidity. So the lipid components can move laterally, endowing the membrane with fluidity, flexibility, and imperviousness to polar molecules. So this property, the property of being fluid and being flexible and being impervious is attributed to the lipid component of the cell membrane. So for example, when we say imperviousness, now that is the inability of polar molecules to penetrate the cell membrane. So example of a polar molecule is water. Another property of the biological membrane is its rigidity. So the property of being rigid is being modulated by the cholesterol component of the cell membrane. So cholesterol tends to stiffen the bilayer, making it more rigid and less permeable. So depending on the temperature, so for example, at low temperature, cholesterol increases the fluidity by keeping phospholipid from packing tightly together, while at high temperatures, it reduces the fluidity of the membrane. So these are the other aggregations, phospholipid aggregations that can form whenever the phospholipids will be subjected into an aqueous solution. So for example, phospholipids can form into a micelle or it can form into a liposome, depending on the location in the body. So these micelles, for example, these are important in the gallbladder, in the liver, in the formation of the bile. Bile is important for the fat emulsification, and for the liposomes, these are important as drug delivery mechanisms in the treatment of certain types of diseases. So the liposomes can serve as a transport mechanism for different drugs in order for it to reach certain target tissues or target organs in the body. So this is the summary of the different components of the plasma membrane. So the main component is the phospholipid. We also have the cholesterol. We also have the integral proteins, peripheral proteins, and the carbohydrates. So before we tackle the absorption of the drugs and the different mechanisms whereby drugs are being transported across the cell membrane, so we have here to consider the different routes of administration that the drug is being that the drug undergoes before it will be absorbed in the body. So of course, there are different routes of administration that are being practiced in veterinary medicine. So we have the IV, IM, intramuscular, per oral, SC, subcutaneous, IP, intraperitoneal, inhalation, and dermal. So for the IV, so since the intravenous route is directed, it is direct to the bloodstream, so it does not undergo the different barriers for absorption. So unlike, for example, when a drug will be administered orally, No, so it will first undergo first the mouth before it will be transported into the stomach, and in the stomach it will undergo different processes in the stomach, no, for it to be absorbed, and it will still, no, pass through the liver before it will reach the systemic circulation. So marami pang dadaanan before a drug will be fully absorbed in the body. No, so unlike for the IV route, pag IV, diretso na siya sa plasma, diretso na siya sa blood, plasma. So drug absorption. So when we say drug absorption, no, that is the transfer of the drug from the site of administration to the bloodstream. And so we have here a diagram of the drug. Then we have here the absorption to the bloodstream. So again, the absorption of a particular drug will basically depend on the site of administration. So that is one of the factors, no, that will determine the rate, no, the amount and the rate of the absorption of a particular drug. For example, for those drugs that are delivered via the IV route, since they are directly delivered into the bloodstream, they have a complete absorption. So they have 100% bioavailability and there is no loss of a drug. But for those drugs that will undergo first the, for example, oral route, they will still, you know, need to be dissolved in the mouth. They need to be dissolved in the stomach, and they need to pass through the liver, you know, before they will totally be absorbed in the body. So there is some loss in the drug components along the way, as contrasted with the intravenous route. So according to the chemical properties of the drug, it's absorbed from the GIT through 4 different mechanisms. So for those drugs that needs to pass through the biological membranes, so these are the mechanisms that they need to undergo in order for them to be absorbed in the body. So for example, a drug would either undergo passive diffusion, facilitated diffusion, active transport, endocytosis and exocytosis, and filtration. So this will basically be governed, or this will basically be depending on the chemical property of the drug. So when we say passive transport or simple diffusion, majority of the drug transport process is executed in this particular type of drug transport mechanism, no, through passive transport or simple diffusion. 99%, no, about 99% or almost all drugs are being transported, no, via simple diffusion. So other types of drug transport mechanism that requires a transport system and that requires energy in order for it, you know, to be materialized are active transport, facilitated diffusion, endocytosis, and exocytosis. For passive diffusion, so this majority of the drugs are absorbed through this mechanism. And the driving force for passive absorption of a drug is the concentration gradient of that particular drug. So when we say concentration gradient, that refers to the drug concentration from the outside, in the outside relative to the inside. So normally for passive diffusion, the concentration gradient is from a higher to a lower concentration, meaning that drug tends to be absorbed from the compartment where the drug concentration is higher to the compartment where the drug concentration is lower. So that is the driving force now for passive diffusion. So passive diffusion does not involve a carrier and does not require energy. So when we say carrier, this is a protein molecule that will serve as a channel, to transport molecules from one compartment to the other. So since for passive diffusion, carriers are not present and the mechanism will also not involve the requirement for energy. So it is said that lipid-soluble drugs readily move across biological membranes due to their solubility and the membrane bilayers. Water-soluble drugs penetrate the cell membrane through aqueous channels or pores. Since the biological membrane is basically composed of lipid bilayers, so only those drugs that are lipid soluble will be able to move across the biological membrane. So this is an example, of an illustration of a passive diffusion. So in this case, uh, some some molecules such as urea and glycerol, and small lipids move with the concentration gradient. So for example, the concentration gradient here is uh, greater as compared to here, so that the driving force will be from the higher to the lower concentration. So in this case, water molecules and water soluble substances, since they are polar, they will not be able to pass through the cell membrane, so that they will be needing certain types of transporters or carriers. So in this case, we have here, the cell membrane, and we have here the presence of a transport protein or a carrier. So this is an integral protein that will transport polar molecules. So for example of our polar molecule is water. Water will not be able to diffuse from or through the cell membrane, so it needs a carrier. So this case here, this is not an example of a passive diffusion, but this is an example of facilitated diffusion, since it requires the presence of a carrier for it to be able to transport molecules. So this is the illustration of the driving force of the passive diffusion or simple diffusion. So the driving force is the concentration gradient. Now we have here two compartments that are separated by biological membrane. So one compartment has a higher amount of the drug molecule. The other compartment has a lower amount of the drug molecule. So for passive diffusion, as long as this particular drug is lipid soluble, so it can readily diffuse from an area of higher to lower concentration through the biological membranes. So this particular transport mechanism follows the fixed law of motion. So the fixed law of motion considers one of the consideration or one of the parameters that are being considered in the fixed law of motion is the permeability coefficient. And this permeability coefficient can be this can be influenced by the lipid solubility of a particular drug. So of course, pag mas higher yung kanyang lipid solubility, the drug will be moving readily across the lipid bilayer. So when the drug will be absorbed, no, in the biological membranes, there are different factors that will affect, no, its absorption. So one of the factors that will affect the drug absorption is the pH of the environment. So it is said that most drugs are either weak acids or weak bases. So when we say acids, or when we say the drug is acidic, or when we say a particular substance is acidic, so that particular substance has the ability to donate or to give off a proton or hydrogen ions. Na yun yung when we say a substance is acidic. When we say a substance is basic, that particular substance has the ability, you know, to take or to accept hydrogen ions. So pag sinabi naman nating weak acid or weak base, when we are going, for example, to put that particular weak acid in a solution, the dissolution is said to be the dissociation or the dissolution of that particular compound is not that complete, no, as compared to, for example, a strong acid or a strong base. So, for example, when a strong acid will be put in a solution such as water, its components will readily dissociate from each other, as compared to a weak acid wherein the dissociation, no, from each other is said to be not complete. So acidic drugs are present in two forms. We have the unionized form and the ionized form. What do we mean by an ionized form? How do we define? Ano nga ibig sabihin ng unionized?
No charge, sir.
So pag sinabi nating unionized, so wala siyang charge, positive or negative. Pag ionized naman, charge, negative or positive. So this is our illustration, no, of our weak acid. So weak acid. So pag HA, that refers to the weak acid. When we are going to put the weak acid in a solution, so since this is an acid, this particular acid will give off, it will donate hydrogen ions. So the hydrogen ions will dissociate from the acid, no, which is this is the conjugate base. So pag sinabi natin conjugate base, no, from the Bronsted-Lowry theory of acid and base, pag sinabi natin conjugate base, now this is an acid wherein the hydrogen ion was removed. Okay. So the H here represents, di ba, yung hydrogen ions, no? The H in this particular HA represents the hydrogen ions. Since the acid, when it will be subjected or when it will be dissolved in water, for example, it tends to give off hydrogen ions. So what remains is the conjugate base. The conjugate base, no, is an acid that is formed after the removal of hydrogen ion. So A minus, no, represents the conjugate base. Conjugate base is an acid that is formed after removal of the H or the hydrogen ions. So the other term for the unionized form is the unprotonated form. The other term for the ionized form or the charged form is the protonated. So pag sinabi natin proton, that has a positive charge. So in this case, in an acid, the proton refers to the hydrogen ion. So pag tanggalin itong hydrogen ion, what remains is the conjugate base, no? Conjugate base A minus. So this one here is the unionized form since it has no charge. This one here is the ionized form since it has charge, no, A minus. For weak basic drugs, now they are present in two forms, an ionized form B or the unionized form BH plus. So when we are going to put a base, a weakly basic drug to a solution, so it tends to dissociate into uh, an ionized form, base, and the ionized form of the and a proton, no, proton. So in this diagram, for example, the unionized or the ionized form of the BH is the form that is protonated. No, protonated because it contains the hydrogen ions. So a drug passes through membranes more readily in its unionized or uncharged or nonpolar form. So only those drugs that are unionized, meaning those drug components that are not charged or nonpolar, will be able to cross the membranes. So for the acidic drugs, now they are unionized in the acidic medium. That's why acidic drugs are absorbed from the stomach with a pH of 1.5 to 3.5. For bases, now they are present in their unionized form in basic medium. That's why basic drugs are mainly absorbed from the intestine. The intestine has a pH of 6 to 7.4. So in the case of weak acids and weak bases, the ability to move from aqueous to lipid or vice versa varies with the pH of the medium. So this particular um, concept is explained by the Henderson-Hasselbalch equation. So this equation can be used to predict the effect of pH change on the drug absorption. So this equation states that for an acid, the pH is equal to the pKa of the drug plus the logarithm of the ionized form over the unionized form. For a base, pH is equal to pKa plus the logarithm of the unionized form over the ionized form. And what do we mean by pKa? So when we say pKa, that is the pH wherein the ratio or the concentration of the ionized form and the unionized form of the drug is equal to 1, meaning that there is a 50% ionized form and 50% unionized form of the drug. So that refers to the pKa of the drug. So basically, for weak acids, they serve as a proton donor. So they have the ability to donate uh, hydrogen ion or a proton in a solution. So ito siya nag-dissociate siya into hydrogen and the conjugate base A minus. So A minus is the protonated form or the ionized form of the acid. For a weak base, it has the ability to accept a proton. So in here, it accepts a proton, so it becomes ionized. So it becomes protonated. So HH equation, Henderson-Hasselbalch equation, is used to calculate the percent ionization of the drug in cellular compartments of different pH. So when we say ionization, that refers to the proportion of the ionized and the unionized form of the drug. So percent ionization. When we say compartments, ano ba yung mga compartments that we refer to? Ano yung ibig sabihin ng cellular compartments? Anong ibig sabihin natin ng mga cellular compartments? How do we mean, or what refers to the cellular compartments? The space within the organism cells. All of the closed parts within the cytosol of a eukaryotic cell. So so those are the definition of our cellular compartments. So in this Henderson-Hasselbalch equation, since we refer to the absorption of drugs, pag sinabi natin cellular compartments, we refer to the different organs in the body. So for example, yung stomach, intestine, the fetal fluid, maternal fluid, placental fluid, yung mga ganun. The different compartments in the body. So, di ba yung ating, for example, stomach, it contains the gastric juice. It has a different pH as compared to the intestinal fluid. The maternal fluid and the fetal fluid. So, yung maternal fluid would have a different pH to that of the fetal fluid. Another is yung mga mga intracellular fluid such as the cerebrospinal fluid in the brain. It has a different type of pH, different pH compared to the blood plasma. So another is the ano pa ba yung other component? The milk. The milk, for example, has a different fluid pH as compared to the plasma. So the pH of the different fluid compartments or the different cellular components, compartments, could influence the degree, percent of ionization of a particular drug. So therefore, we can say that the absorption of a particular drug is not only influenced by whether that particular drug is acidic or whether that particular drug is basic, but it could also be influenced by, number one, its pH, whether that particular drug is acidic or basic. Number two, the pH of the environment, whether the environment is basic or acidic. So understanding how changes in pH alter the ionization of the drugs is very important since unionized drugs cross the membranes. So again, when we say pKa, that refers to the pH at which half of the molecules are in the ionized form and one half of the molecules are in the unionized form, meaning that the proportion of the ionized to the unionized form of a particular drug is equal to 1. So in the Henderson, pH and drug concentration are log-based scale. So every point difference in pH is tenfold difference in the drug concentration. So 10-point fold no difference that is this means that the concentration of the drug, the percent ionization of the drug, whether the drug is ionized or unionized, is expressed in tenfold. Now, depending on the increase or the decrease of the pH. So, for example, one level increase in the pH of acidic drug, for example, or 1 degree change to the pH of a particular compartment would lead also to a tenfold increase in the ionized or the unionized form of a particular drug. So for the degree of ionization or the polarity, so only an unionized drugs diffuses across the membrane. Hence, this is an important factor. So non-polar drugs are lipid soluble. Polar drugs are water soluble. They can't absorb from the biological membrane. So only the unionized forms of the drug or the uncharged drug can pass through the membrane by passive diffusion. By controlling the pH of the solution and the pKa of the drug, you can control the rate at which the drug is being transferred. So in this example, we have here two examples or two types of drugs. We have the drug that is non-ionized and the drug that is ionized. So since this particular molecule here is non-ionized, it is non-polar, so it is lipid soluble, the particular drug can readily pass through the cell membrane. For those drugs that are ionized, polar or water soluble, so they are trapped within in this side because they will not be able to pass through the cell membrane. So the pH of a particular compartment or the diffusion of a drug would also be influenced by, again, the pH. And for monogastric animals, of course, the pH in the stomach of a monogastric animal is different from the pH of the stomach of a ruminant. So for monogastric animals, since the monogastric animals has a low pH, weak acids such as aspirin with a pKa of 3.5 tend to be better absorbed from the stomach than weak bases because of the acidic conditions. So in ruminants, the pH varies with the feeds and the pH is often not low. So for ruminants, they usually have higher pH or a pH that is more to the basic side as compared to the monogastric animals such as dogs, wherein the pH of the stomach is acidic. So for those drugs that are weak acids, they tend to be better absorbed from the stomach because when we are going to place an acidic drug to an acidic environment, that drug would be unionized. It will be or the proportion of the unionized form of the drug would be higher as compared to its ionized form. So pag mas malaki yung the unionized form of the drug, no, so that particular drug will be readily absorbed in the stomach. So this can be, we can conclude, no, that when we are going to place an acidic drug to an acidic environment, that particular drug will be absorbed. When we are going to place a basic drug to a basic environment, there will be absorption. But when we are going to place an acidic drug to a basic environment, so there will be less absorption or no absorption. So that is also true for a basic drug. For weak bases, they are poorly absorbed from the stomach since they exist mostly in the ionized state. They have low lipid solubility because of the acidic conditions. So they are better absorbed from the small intestines due to the higher environmental pH. So this is another illustration of the passive diffusion of our weak base and our weak acid. So when we say again, pKa, that is the pH wherein the proportion of the ionized to the unionized form of the drug, whether it is a weak base or a weak acid, is equal to 1. So for example, when we are going to place an acidic drug in an acidic environment, so this is the pH of the environment, pH 3, 4, 5, 6, 6.5, 7, 8, 9, 10, 11. So those are the pH of the environment. So when we are going to place an acidic drug in an acidic environment, the proportion of the non-ionized form of the drug will predominate so that the drug will be absorbed. When we are going to place an acidic drug to a basic environment, the proportion of the ionized form of the drug will be predominating so that the drug will be trapped in this alkaline or basic environment. When we are going to place a basic drug in an acidic environment, the pH will be, the drug will not be absorbed because the ionized form will predominate. And when we are going to place a basic drug in a basic environment, it will be absorbed because it is present in its unionized form. We also have here another diagram showing the absorption of our acidic drug. So this particular drug has a pKa of 5. So meaning that at the pH of 5, the proportion of the ionized and the unionized form of the drug is equal to 1. And these are the different body compartments. We have here the membrane, the cellular membrane, that separates these two body compartments. One body compartment has a pH of 3, so it is acidic. One body compartment has a pH of 7, so it is towards the basic side. So what happens for an acidic drug when we are going to put it in an acidic environment with a pH of 3? So when we are going to put an acidic drug in an acidic environment, what happens to the unionized and the unionized form of the drug is that the ratio of the unionized So the ionized form becomes 1 is to 0.01. So the difference here is, yan yung difference nila. A factor of 100, no? Factor of 100, no? Because, ano siya, no? 0.01, 0.01, then 1. So dalawang zero, so factor of 100. Ito naman, when we are going to place an acidic drug to a basic environment, so what would happen is the ionized form of the drug, of the acidic drug will predominate. So dito siya, no? This is the ionized form, no? The conjugate base, and this is the acid form, this is the ionized form. So makikita natin dito na ionized kaysa sa unionized. So therefore, pag mas marami yung ionized form of the drug, there will be drug fluid compartment. It will not diffuse across the membrane, so it will be trapped in this fluid compartment. So from the diagram, we can say that each unit decrease, or each unit that we are going to move, the pH of the environment towards the acidic side, the proportion of the unionized form of the drug will also increase by 10. So for example, when we are going to place this particular acidic drug with a pK of 5 to a pH of 4, the proportion of the unionized, so the ionized form will be 1 is to 0.1, meaning that there is an increase in the unionized form of the drug tenfold. So further increasing the pH towards the acidic side, so meaning pH of 3, then pH of 2, will also increase the proportion of the unionized form of the drug by 10. By 10. And when we reach, for example, a pH of 4, when we are going to put this drug at a pH of 4, so mag-increase ngayon yung unionized form of the drug by 10. Further decreasing the pH to 3, mag-increase naman siya by 10. So the difference here now is 2 levels, pH of 5, then pH of 3, so that is equal to 100. So 10 times 10, 100. So 2 level, each unit increase or decrease towards the acidic side will also increase the level of the ionized or the unionized form of the drug. So dito naman, di ba yung pKa ng ating drug is 5? When we are going to increase the pH towards the basic side, so halimbawa when we are going to increase it by 6, the proportion of the ionized form of the drug will also increase by 10. Pag ginawa natin ito siyang 7, so magiging 100. Pag ginawa natin ito siyang 8, yung pH, this will be 1,000. Pag ginawa natin ito siyang 9, this will be 10,000. 10, 100,000. 11, 1 million, and so on and so forth. Pag ito naman, yung pH, yung acidic drug, with the pKa of 5, no, ginawa natin siyang, yung pH dito is ginawa natin 4. This will be 0.1, no, 0.1. And pag ginawa naman natin ito siyang 3, so 0.01. Pag ginawa natin ito siyang 2, this would be 0.001. And so on and so forth. So meaning that when we are going to increase, or in this case, we are going to decrease the pH of the environment, no, for an acidic drug, there will also be an increase in the proportion of the unionized form of the drug so that there will be absorption. When we are going to increase the pH of the environment where an acidic drug is being administered, so what will happen is there will also be an increase in the proportion of the ionized form of the drug so that the drug will not be absorbed and it will be accumulated in that particular body compartment. So I think we'll end here. And do you have questions in this figure? May tanong ba kayo dito sa figure natin? This particular figure. May tanong kayo dito sa figure?
None, sir.
Okay. So basta yung ating ano lang dyan, yung ating key points there is pag ionize yung drug, it will not be absorbed. Only the unionized form or the uncharged form of the drug will be absorbed. And we also have to, of course, know what is pKa. That is the pH of the environment where the proportion of the ionized and the unionized form of the drug will be equal to 1. So yun lang. No? Pag nilagay natin yung acidic drug sa acidic environment, it will be absorbed. Acidic drug to a basic, it will not be absorbed. Basic to basic, it will be absorbed. Basic to acidic, it will not be absorbed or it will be trapped in the environment. Kaya may tinatawag tayo na ion trapping no mechanism wherein the ions or the drug could either be trapped in a specific environment depending on the pH where that particular drug is being placed. So if wala nang tanong, so we'll just continue this next meeting. So good morning to everyone and see you next meeting.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Sir, excuse me po. Anong
Hello.
oras po sa Friday mag-quiz?
Sir.
Friday, kayo mag-decide. But preferably siguro mga morning kayo ang... Kasi meron din akong isang long exam na 4th year. And pinili nila yung hapon. So siguro pwede kayo morning. Friday ba kayo or Thursday? Friday ba kayo? Friday?
Yes po, yun po yung sabi niyo,
Friday.
October 8.
So, pwede naman kayong Thursday or Friday. Pero kung Friday kayo, so morning morning para yung 4th year ko, na isa kasi may 4th year din ako na mag-exam din sila, so hapon naman sila. Okay lang, morning?
Okay po, sir.
Okay.